Welcome back to BinaryBio. Today we are going to solve the shortest superstring problem, a fundamental challenge in computational biology. Imagine you shred a book into overlapping strips, and your job is to reconstruct the original text. That's exactly what genome assembly is. DNA sequences don't read whole chromosomes, they produce short fragments called reads. Because the same idea underlies how scientists reconstruct entire genomes from sequencing data. Today, we'll solve the simplified version of this problem, reconstructing a chromosome by building a shortest superstring. A superstring is simply a larger string that contains all smaller strings inside it. For example, if we have reads ATTAG, TAGAC, and GACCT, one valid superstring is ATTAGACCT because it contains all three reads. But we want the shortest one because in biology we assume the simplest reconstruction is most likely. If we simply glued reads end to end, we'd get a long, redundant sequence. By maximizing overlaps, we recover the most likely original genome. Normally, the shortest superstring problem is very difficult. Without constraints, we would have to test many possible read orders, and the number of possibilities grows exponentially. But this specific problem gives us two guarantees. Valid overlaps are longer than half the read length, and there is exactly one correct reconstruction. That means the reads form one clear chain rather than a branching graph. Our job becomes much simpler. We just need to discover that chain. Let's walk slowly through an example, and this time we label the reads with IDs so it's easier to follow. Suppose we have four reads. Each read has length 10, so half the length is 5. That means valid overlaps must be at least 6 bases long. Now we compare reads pair by pair. Read 1 ends with this string, which matches the beginning of read 3. That's a 7 base overlap, so we record a connection. Read 1 to read 3. Next, read 3 ends with this, which matches the beginning of read 2, so we record read 3 to read 2. Then read 2 ends with this, which matches the beginning of read 4 with an overlap of 7. So we record read 2 to read 4. Now we determine the starting read. The start must be the read that no other read overlaps into. Read 3 has a predecessor, read 1. Read 2 has a predecessor, read 3. Read 4 has a predecessor, read 2. Only read 1 has no predecessor, so that must be the beginning of the chromosome. Now we merge them step by step. We start with read 1. We add read 3 next. Since they overlap by 7 bases, we don't repeat that shared section. We only append the new part, CCG. Next, we add read 2. We append only GAA. Finally, we add read 4. The overlap is 7 bases, so we append only ATA. The final reconstructed chromosome is this. We just turned 4 separate fragments into a single continuous DNA sequence. Now let's see how this translates into code. This imports BioPython's FASTA parser. Instead of manually reading lines and stripping the greater than symbol, we use a built-in parser. The function reads the FASTA file and returns a list of seq record objects. Each record contains two main pieces of information: the record ID, which is the read name, and the record sequence, which is the DNA string. So after this step, we simply have a list of reads we can iterate over. Next comes the overlap function. This function answers a very specific question. How much of the end of one read matches the beginning of another read? We can't overlap more than the shorter read, so we start with the largest possible overlap and work downward. As soon as the suffix of the first read matches the prefix of the second, we return that length. This function is really the core comparison engine of the algorithm. Then we build the overlap map. Here we compare every read against every other read. We create two data structures. The first stores connections between reads, meaning which read should follow another in the genome. The second is a set that tracks which reads already have a predecessor. If a read appears in this set, it means something must come before it. During this process, we skip comparing a read with itself and we convert BioPython sequence objects into plain strings so they're easier to compare. We only keep overlaps that are longer than half the read length because the problem guarantees that valid neighbors must overlap strongly. When we find such a match, we record that one read comes before another and we mark the second read as having a predecessor. After building this map, we find the starting read. The first read must be the one that no other read points to. So we simply look for the read that never appears in the incoming set. That read becomes the beginning of our chromosome. Finally, we assemble the genome. First, we build a lookup table so we can quickly retrieve the DNA sequence for any read ID. We begin with the starting read and then follow the chain of overlaps. Each time we move to the next read, we append only the non overlapping portion so we don't duplicate sequence. We repeat this until there is no next read left, and the result is the reconstructed genome. This works perfectly here because overlaps are strong, unique, and form a single path. In real genome assembly, data is noisy and genomes contain repeats. So modern assemblers use more complex graph models like De Bruijn graphs. But this simplified version captures the core idea behind how genome assembly works. One quick note on efficiency. If we have n reads of length L, we compare every read with every other read, giving about n squared comparisons. Each comparison may check overlaps up to length L, so the overall runtime is roughly proportional to n squared times L squared. That's fast enough for small datasets like Rosalind problems. If this video helped you understand genome assembly, subscribe to BinaryBio and stay tuned for more bioinformatics algorithms. See you in the next one.